Okay, thank you very much. I'm delighted to be talking to you about something that might not immediately seem that relevant to chemistry. I thought just before I leap into my talk, I'd just give you a little bit more background about myself, in the sense that at school, I could never decide between the kind of humanities subjects and the science subjects. So I took biology, chemistry, classics and French. And I have to say that by pursuing chemistry, I've actually not had to choose between the two. So as Saskia said, I actually studied archaeology and chemistry at university together. And then what my job involves now is basically using chemistry as a tool, essentially various analytical chemistry tools, in order to understand more about the past. So just to sort of tell you that you can do it, you can combine kind of two loves that might seem quite disparate, but actually really complement each other. And I think it's really helped me to be able to understand much more about the past, which is actually what I'm going to tell you about now. So I want to start my talk with this picture, because this is basically what I and other archaeological scientists are trying to do. We're trying to essentially reconstruct a picture a bit like this of what life was like in the past. How were people living? Where were they living? What were they eating? What did the landscape look like? How did they interact with their environment? How did they manage their kind of their livestock and their crops sustainably? How did that last through long periods of time? But the actual picture that we generally have about the past is a lot more like this. So your eyes haven't gone funny, it's much more blurry. We have a sort of a general idea of the shapes of the houses people lived in. We might have an idea of the types of foods people were eating, but nothing particularly concrete. And so I think what chemistry can do is actually provide sort of little snapshots, little clear areas within this picture, within the archaeological context. So we still need to excavate, we still need to dig things up and get that general picture, but we can get these kind of clearer insights into various aspects of life in the past using chemistry as a tool. And so I've kind of shown a few of these areas that I particularly have been working on. There's lots of other areas. And actually today I'm going to focus particularly on reconstructing what people were eating, so their diet, and then also a little about mobility. So where were people moving, about 1,000 years ago? Where were they coming from? How connected were they? So just give you a little insight into some of the things I do amongst everything else. Okay, so when we talk about diet, as archaeologists, what do we generally have at our fingertips to understand what people were eating? Well, traditionally, we pick up animal bones from sites, so these get preserved in the soil. Then we can identify what species. We can say, okay, they were eating beef, they had cattle, they had sheep and goats, they had pigs. And then we may also find charred crop remains. So we might find wheat, barley seeds, we might find some peas, some beans. And then if we're closer to the sea, we might find shells. So this will give us an idea of what kind of species people were eating, but it doesn't really give us an idea of how important these different types of foods were to people. They're basically the rubbish that people discarded and then tend to get preserved in the soil, and then we excavate them. But that's not necessarily telling us about how important they were and what people were actually eating. It's just what they were throwing away. So using chemistry, we can actually add a few materials that we also find on archaeological sites that will help us get a better idea of what people were actually eating. So I'm not going to talk about pottery residues. We can actually look at the fats that get absorbed into pots that we find in archaeological sites, and we can find out about what types of fats, whether it's dairy fats, meat fats, that people were cooking. What I'm going to focus on today is actually the human bones and human teeth that also can be preserved on archaeological sites. So the kind of the cemeteries or even within settlements. So it's the actual people themselves. We can study their bones, their teeth, to understand what they were eating. You might wonder, well, how on earth do we do that? Well, it basically all comes down to this premise: you are what you eat. So basically the nutrients that are in your food, and if we're thinking about chemistry, the elements, the carbon, the nitrogen, the oxygen, the hydrogen, that's in the foods that you eat is used to build your proteins, basically the tissues of your body. And so we're almost like detectives. If we start off with the bones and the teeth of the people, we can go back to reconstruct, well, what were they built from? What were the building blocks? What were the sources of the protein and the minerals that we're actually excavating from archaeological sites? Well, how do we actually do that? What I actually focus on is isotopes. So I'm not sure how familiar you are with isotopes, so I'm just going to give a quick kind of overview of what they are. So essentially, isotopes are different forms of the same element. So what makes an element now, and what makes carbon, nitrogen, is the number of protons in the nucleus. So carbon will always have 6 protons, but then different isotopes have different numbers of neutrons. So the common isotopes of carbon are carbon-12, carbon-13. So they contain either 6 neutrons or 7 neutrons. And there's also carbon-14, which you might be familiar with. And again, archaeologists will think about immediately, because this is radiocarbon. So this is actually not stable. It decays and it allows us to date things. So measuring the amount of radiocarbon in a sample allows us to know how old it was. I ignore that because actually it doesn't tell us about diet, but just so that you know there is this other isotope. And then for nitrogen, similarly, what makes nitrogen is that it has 7 protons, but then you can have different amounts of neutrons. So what we do is we use mass spectrometers. We use isotope ratio mass spectrometers, so they can actually measure the different relative amounts of different isotopes. And that will give us an isotope value. And that can basically tell us something about where that carbon comes from, where that nitrogen comes from, and tell us about diet. And the reason why we can why this works is because basically isotopes of different masses behave ever so slightly differently. I mean, you might think the difference is so tiny, but actually it does make a difference on a kind of atomic level. So heavier isotopes, as you may well expect, logically, will tend to react more slowly than lighter isotopes in anything that involves energy, in kinetic reactions. So if that's diffusion of different gases, if that's evaporation, so from liquid to gas, but also in enzymatic reactions. So anything involving kind of biology, you've got enzymes that are converting one reactant to a product in the body. You tend to get the lighter isotope ending up in whatever is created, and you get the heavier isotope left behind. So this is my very, very clumsy, but essentially it is like they just move, the lighter isotope just moves faster than the heavier isotope. And so we can, by measuring the relative amounts of the 2 different isotopes in a sample, it will both reflect the original source, the original proportions of those isotopes from wherever that came from, but it can also reflect the different processes that take place. So if you think when you eat something, it has to be digested, it has to kind of enter the body, it has to be metabolized, it has to be converted into sort of different amino acids, for example, that are then used to build the proteins in the body. All of those processes, every sort of reaction that involves an enzyme will result in different proportions of the lighter isotope entering them. And so we can almost work out what's happened to those, to the carbon and to the nitrogen from being eaten to being incorporated into the body's tissues. Just to show you that, so I'm going to focus today carbon and nitrogen, because they are the kind of building blocks of proteins. I'm also going to talk to you about oxygen and sulfur. But just to show you the periodic table, there are a lot of elements that have multiple isotopes. So I always show this to my master's students because I just think it's exciting. It's showing you that basically all of the elements that are shaded in grey have multiple isotopes. but we don't use them yet as traces for anything. So it's kind of, there's this world out there where we could potentially measure the relative amounts of isotopes of these elements, and maybe they will tell us something. Iron, for example, shaded in green, it's rarely used, but it does reflect certain things about the metabolism of the body. Zinc isotopes have actually been used to identify individuals with breast cancer versus those that don't have breast cancer. Calcium can actually indicate things like how important dairy was in people's diets. So there's lots of potential. So just by focusing on basically on this little kind of sideways L, I'm being quite focused, but there is quite a lot of exciting opportunities for future work for budding chemists who want to do that. Okay, so I mentioned we have human bones, we have human teeth. So what do we actually analyse? Well, we actually tend to focus on the protein that is preserved in the bone. So that is the collagen. So we all have collagen. We have collagen in our skin, in our nails, in our hair, in our bones. And this actually tends to be preserved. It can be preserved for, depending on the conditions, up to about 40,000 years, maybe even a little bit longer. I don't work on such long time periods. I tend to work up to about 10,000 years ago. But you still have protein preserved in the bones. And that protein contains carbon, nitrogen, and a very, very small amount of sulfur. It's basically built up of amino acids. And the thing about bones, so when I say that we can reconstruct diets, that's true. But it's very much an average diet. So we're not picking apart individual meals. We're not sort of getting little snapshots of what people were eating maybe on a day-by-day basis. It's actually representing an averaging of the source of the carbon, nitrogen, and sulfur that's used to build those bones over for long bones, for example, of potentially a few decades, depending how old the individual is. It basically depends on how fast your bones are being kind of remodeled. So it's much faster during adolescence, and then that slows down as you get older. But different bones will turn over, will be remodeled at different rates. So my femur remodels much more slowly than ribs, for example. So we can actually compare the isotope values of long bones and ribs, and get different sort of snapshots of diet. And so we can look at sort of the last couple of decades of someone's life and what their diet looked like compared to the last, say, 5 years of their life. And similarly, different types of bones will turn over at different rates. So again, if you think of the femur, you've got that sort of the dense shaft of the femur. That will turn over much more slowly. That's the cortical bone than the kind of the spongy ends. So again, we can kind of compare. So bones provide us a sort of a long-term average, really, of say, the last decade or so of someone's life, whereas teeth, they actually form during childhood, and then they do not remodel again. So they provide actually very discrete sort of snapshots of childhood diet over the period of the tooth formation. So again, with teeth, we have the collagen, which is in the dentine. That's the kind of the root of the teeth. The sort of, that's where if you get a cavity, it's because the hole's gone through to the dentine and it hurts. So by measuring the isotope values in the dentine, again, we're looking at the protein, so we can get carbon, nitrogen, and sulfur isotope values. But then we've also got the enamel, so the white sort of covering to the teeth. And this is basically the mineral portion of the teeth. We also have this in bone, but it's a lot more likely to degrade in the soil. So we don't tend to trust, it can get contaminated. Whereas enamel it's like a very strong or crystalline mineral. So we can actually measure the isotope values of the enamel. And that doesn't contain protein or very, very small amounts. So for that, we're basically concentrating on carbon and oxygen. And I'll tell you a little bit. I've got a case study where we've actually measured these isotope values of people. So the other nice thing about teeth is, as you know, they form at different times. They erupt at different periods. So we tend to focus on the molars. So these are the teeth, at the back of your mouth. The first molar will basically come out, will erupt at about 10 years old, but it's been forming since birth. So it basically starts forming under your gum line and only just comes above the gum at about 10. Some of you might not actually have your wisdom tooth, your third molar yet, but again, it's been forming since about 8 or 9 years old. Sometimes we actually find them in sort of archaeological skeletons. They haven't come out yet, but it has been forming. And so if we sample sort of take tiny samples down the root of the teeth and all 3 molars, we can actually see how someone's diet changed between birth and early 20s year of age. So if you think about the sort of changes that you might have had in your diet, you start off just solely on milk, so maybe breastfed, and then you will shift to solid food. Maybe there'll be some changes. If people moved from a coastal site where they might have had a lot of access to a lot of fish, but then they moved inland, then their diet might have changed. And similarly with the enamel, it can pick up, with the oxygen, it can pick up where they were, whether they moved during their childhood. So with bones and teeth, we have these ways of accessing what people were eating both during childhood and then also for, say, the decade before they died, depending on how old they were. Okay, so what can we actually find out about diet? Well, carbon isotope values are very good at separating between marine resources, so things that come from the ocean, fish, marine mammals, maybe seals, and terrestrial resources. So that would be your cattle, your sheep, your goat. And this is basically because of the source of the carbon. So terrestrial plants photosynthesize, and that basically, that process, because there are enzymes involved, you get you end up with a lot more of the lighter isotope of carbon in the leaves of plants like wheat and barley that we tend to eat, whereas carbon in the oceans is a mix of that, but it also comes from dissolved carbonates on the kind of ocean floor, and they tend to have higher carbon isotope values. So we get this clear difference between the carbon isotope values of marine fish and any sort of plants and animals that are living on land. So this was kind of established decades ago, but very simply, we get this difference. The isotope values of C3 plants, these are the majority of plants that grow in kind of temperate Europe, have much lower, you see very negative carbon isotope values, about -27 per mil, compared to marine fish, which are more like -12 per mil. Now, I won't talk about C4 plants. These are things like maize, millets, sugarcane is a C4 plant, because we eat them today, but they tend to grow better in sort of tropical regions. And I'm going to talk to you more about a British kind of example not far away from here. It's a bit more difficult to distinguish between C4 and marine. We have to use different ways of doing that. And then nitrogen, this is actually more about where you are on the food chain. So plants have the lowest nitrogen isotope values, and then as you go up the food chain, you basically excrete the lighter isotope of nitrogen, and so your body becomes enriched in the heavier isotope. And so you can see this example from human hair. Vegans who are eating a purely plant-based diet have got lower nitrogen isotope values than both vegetarians and omnivores, because even vegetarians are eating animal products. So if they're eating dairy products, if they're eating eggs, then they will still be they are still eating animal proteins. But actually what's really nice is that there's a difference depending on how much of those animal proteins people are eating. So people who are only eating occasional meat or occasional dairy products will have lower nitrogen isotope values than people who are eating them daily. And so again, this is giving us a kind of indication of how important plant versus animal protein was for people in the past. So this is kind of the go-to slide. This is kind of the framework that we use for interpreting the carbon and the nitrogen isotope values that we measure in the bones and teeth of people that we dig up on archaeological sites. So on the x-axis, we've got the carbon isotope values. So the carbon isotope values towards the right-hand side, so they're higher. It's a bit confusing because they're all negative, but they tend to reflect marine resources. Whereas on the left-hand side, the lower carbon isotope values, that's more terrestrial. And then on the y-axis, we've got the nitrogen isotope values. As you go further up the y-axis with the higher nitrogen isotope values, that's basically further up the food chain. So we've got our vegans below our omnivores. But then also what's really nice is that aquatic food chains, so that's our fish from the ocean, but also fish from rivers, they are longer than the terrestrial food chain. If you think about terrestrial, you've got plants, you've got herbivores, you've got carnivores, and that's it. You don't really have anything that eats. You might have an apex carnivore that eats other carnivores, but you don't have any. You can't go any further. Whereas in marine ecosystems, for example, you've got the plants, then you've got the zooplankton, then you've got small fish that eat the zooplankton, then you've got bigger fish that eat the small fish, and then bigger fish that eat the slightly bigger fish. So you can go quite a long way up. And if you're eating seals, then they're actually eating they're really high up the food chain. So you can get really high nitrogen isotope values. So that's another good way of identifying how important marine foods and fish were for people in the past. So this is, actually every year we have a master's course in archaeological science, and we have a practical where the students will basically cut one fingernail, and we will measure the carbon and nitrogen isotope values of that fingernail. And so those values are shown in the green and the orange dots. And this is just to show you we've comparing their carbon and nitrogen isotope values to people who lived, so Neolithic is about 5,000 years ago. So Ascot under Wychwood is only about 15 miles north of here, in the Cotswolds. So these were people living 5,000 years ago, not too far away. So they're in the middle of the country, basically eating a terrestrial diet, similar to our master's students. You can see that there are two vegetarians in green. They obviously eat less kind of animal products than their colleagues who tend to eat some meat. And just to show you that that's very different from people living also 5,000 years ago, but on Oronsay, which is in the Inner Hebrides in Scotland. And they're on an island, so they were obviously eating a lot more fish and marine resources. So there's a really, really big difference. But even within these kind of groups, we can pick out people who were probably eating a little bit more meat, or a little bit less meat, or a little bit more fish, and a little bit less fish. Sulfur can also tell us a bit about fish. So the sulfur isotope value of the ocean is pretty uniform at about 20 per mil. So if we measure the sulfur isotope values of someone who eats a lot of fish, then we'd expect them to have a sulfur isotope value of about 20. But you also, it's not just about the diet, sulfur can also tell us about where people were living. So you get what's called a sea spray effect. Basically, the spray from the ocean will also have a sulfur isotope value of about 20, and that gets blown across the land. So if you notice that the kind of basically Ireland, the west coast of the UK, tend to have sulfur isotope values, those red colours, of about 20 per mil. That's because the prevailing winds coming from the west is blowing that sea spray over the land and increasing the sulfur isotope value of the plants, which then people eat, animals eat, so they will also have higher sulfur isotope values. And then as you get further inland, kind of in continental Europe, the sulfur isotope values actually are more reflective of the kind of underlying geology. So plants are taking up sulfur from the soil, and that's what changes their sulfur isotope values. So we can use it as, it gives us some indication of how important marine fish were, but also how far people were from the coast, also where they were living in general. And oxygen, so if you remember oxygen, we can measure the isotopes in tooth enamel, and that is reflected again of where people were. It's essentially down to the source of the rainfall. So again, people will drink water, and generally in the past that would have come from streams, rivers, lakes, but the oxygen isotope value of that rainfall depends on the source of the rain. So ultimately it comes from the ocean, but then it's how far that rain has travelled to get to the place that the people are drinking it, and the plants that people are eating are getting their water. So it's basically influenced by the distance from the ocean, but also by the temperature. So cold air can't hold as much water, so you tend to have the heavier isotope. If you think about it again, it's kind of logical. Heavier isotopes will fall faster, so you get the heavier isotope falling sooner when the water evaporates from the ocean, and as you get further away from the ocean, you're only left with a lighter isotope, which means you get these lower oxygen isotope values. But similarly, in colder areas, you also get the lower oxygen isotope values. As you go further north, you'll see these darker colours are in the far northern latitudes, so kind of Scandinavia. That's also because it's colder, and so you've got less of the heavy isotope, more of the light. And again, with altitude, it gets colder, and so you've got these lower oxygen isotope values in, you can probably spot the Alps, and also the Pyrenees between Spain and France. So again, we can get an idea by measuring oxygen isotope values in people's teeth, where they might have spent their childhood, and then think, well, is this where they were buried? Is it matching the oxygen isotope value of the water where they're buried, or is it not? Which is telling us they must have moved, and then we can have an idea of where they may have moved from. And just to give you an example, so this is actually the British Geological Survey. They do quite a lot of isotope analysis, and they have got this map where, so even within the UK, there's a difference in the oxygen isotope value of people's tooth enamel between the east and west. And so again, if we sort of dig someone up from Birmingham, and they've actually got a high oxygen isotope value in their tooth enamel, then we can be pretty sure that they didn't grow up in Birmingham. They must have lived somewhere, maybe from further west in the UK, but actually there's lots of other areas of Europe where they could also have had these higher isotope values. So it gives us an idea of kind of whether people were had lived locally to the area in which they died, or whether they'd actually come from somewhere else. Okay, so I've given you a kind of overview, and it probably feels a bit abstract, and you're thinking, well, okay, how does this actually work in practice? So what I want to do is talk you through a bit of a case study of an actual archaeological site, and what we found out, just a little bit about some of the people, what we found out about their lives, about what they were eating, about where they might have come from. So this is actually, I know maybe not many of you are actually local to Oxford, but this is an example of somewhere pretty close to here. So we've got Oxford here in the White Star, and only about, probably about 6 miles away south, there's this very small town called Milton. Basically, they were building a new housing estate, and when that happens, archaeologists come along and they basically check while the diggers move in that there's nothing coming up that might be interesting. And in this case, there was. So there was basically an early medieval cemetery. There were about 50 graves. So they halted the construction work. Archaeologists came in and excavated properly, recorded the cemetery. They also excavated all of the human remains, and they've stored them. They're stored currently in Swindon. So each of these kind of little ovals with kind of marks inside, these are individual skeletons, they're individual graves. As I said, about 50 people from about 1,200 years ago. So this is the early medieval period. And this is a really interesting cemetery because it's actually located, so again, it's the red star, on the border of 2 Anglo-Saxon kingdoms. So there's Mercia and then there's Gavissa. And they were kind of two different kingdoms, they had two different kings, didn't like each other very much. There were lots of battles, particularly on the border of those two. So it's a particularly interesting place in terms of understanding what people's lives were like, how much mobility there was. It's also just unusual because what they found was there's quite a lot of men buried there, and relatively sort of middle-aged men, so at the prime of their life, which is not very common. It's usually sort of young children, maybe women, older people, not middle-aged men. And also quite a lot of men, in particular, with healed weapon wounds. So this is, I haven't put anything too gribbly in, but this is actually a cut mark from a sword in someone's head, but it's healed, so they survived this injury. And so one sort of idea was that actually this was a place where people could come and receive healing. There's another woman who, so she was about 45, like 40s, early 50s maybe when she died, but she had pituitary dwarfism, so she was very, very small, but she also, the signs that she had cancer that had actually shown up in her bones, which means it was very, very advanced. And also her jaw was essentially immobile. So she wouldn't have been able to chew food. So she must have had, people must have prepared the food to allow her to live to the age of, in her 40s. So this suggests that it was kind of a special place where people were caring for others, and maybe a place of healing. So it's just understanding a bit more about, well, who were these people? Where did they come from? Did they have special diets or not? So what we did was we measured the carbon, nitrogen and sulfur isotope values of their bones to give us an idea of what they were eating as adults, so kind of that last 10 years of their life. We also measured the sulfur can also give us an idea of maybe how close to the coast they were living. The oxygen isotope values of their tooth enamel can tell us about maybe where they were born, at least whether they were born local to the site or whether they actually came from elsewhere. And then the carbon in their enamel and in their dentine and the nitrogen in their dentine can also give us an idea of what their diet was like during childhood and whether it changed. I'm just going to talk you through two people that we found particularly interesting and kind of what we learned about them. So just a general overview of, so this is actually everyone from the site, just their bones. You see the carbon on the x-axis, the nitrogen on the y-axis. They've all got relatively similar carbon and nitrogen isotope values, so actually nothing's particularly interesting. There is one man who's got higher nitrogen isotope values, so maybe he was eating a bit more fish than anyone else. There is this a single cow from the site, so we compared them, a herbivore, with the people, and so you can see that the people were obviously eating some meat because they've got higher nitrogen isotope values than the cow. This 1092, this is the woman with who actually would have struggled to chew, and you can see she's basically in the middle. So she's eating a very similar diet to everyone else, but presumably kind of prepared in a way that she was able to eat it. And they have very similar isotope values, so therefore probably very similar diet to people who were buried in a kind of relatively nearby cemetery of a similar age. So they're not sort of sticking out in terms of, even this is actually quite an unusual community or an unusual position, unusual in terms of people needing care. Their diet doesn't seem to be particularly unusual, so not particularly interesting from that perspective. But then we look at the sulfur. So now the nitrogen isotope values are on the x-axis, and the sulfur isotope values are on the y-axis. So if you remember the person, this 1108, who's got these high nitrogen isotope values, if that was because they were eating a lot of marine fish before they died, then you'd expect them to be closer to the sort of marine end of the sulfur, and they're not. So it suggests that maybe it's more to do with eating a bit more meat than everyone else. Whereas actually we've got two men, again, who've got higher sulfur isotope values than other people, and maybe that means that they, at some point in the kind of decade before they died, they were either living closer to the sea all were eating some marine fish, which the others weren't. What's also really nice is that we've got an idea of what the local sulfur isotope value is for the region, for Oxfordshire. And all the green diamonds are children from the site. They all plot within that. So it suggests that they all kind of grew up in the local area, which makes sense. But about half of the adults don't have sulfur isotope values, and it suggests that they had a local diet for the entire sort of period before their death. So that's pointing to quite a lot of mobility, which actually is what we expect for this time period. There was a lot of movement in the kind of early medieval period. People didn't necessarily grow up and die where they were born. They didn't die where they grew up. So this is the kind of the mobility side of things. So now this particular man, so relatively young, kind of in his early 20s, he was the one who had this healed injury to his skull. So he'd obviously been in a fight at some point, but he had healed before he died. So he didn't die of this particular injury. So we measured the oxygen isotope values of his teeth, so 2 of his teeth, which gives us an idea of kind of where he might have spent the first 6 years of his life. Now his oxygen isotope values are too low to have been born in Britain. So that's saying that he's come from the continent. And it's suggesting that he's either come from somewhere a bit further north, which might fit with the Anglo-Saxons, who know much but Scandinavia, or from somewhere at a higher altitude, further south. So we sort of thought, oh, well, Scandinavia, that makes sense. But actually, there's some DNA work that's been done and actually puts a lot of these people more towards the sort of the southern Europe. So it's more likely, actually, that he probably came from a sort of mountainous region in the sort of mainland south-central Europe. He also is one of the people who had sulfur isotope values of his bones that are not local. So he basically couldn't have spent the last sort of 10 years of his life local to the site or somewhere that has similar sulfur isotope values. So he likely moved to the site, not that long before his death. So maybe he came to fight in this particular kind of during these battles between these kingdoms. The other thing that's kind of interesting is these are the isotope values at different places down the root of his teeth. So on the bottom it shows the age. This is kind of an estimate of when those particular parts of his tooth formed, so between 8 and 14 years of age. His teeth had signs of what's called enamel hyperplasia. This is basically when the enamel stops forming because of extreme stress, usually because people don't have enough to eat. So during childhood, he had periods where he just didn't have enough. There was periods of starvation, and that also matches with the carbon and nitrogen isotope values. You see this kind of this sort of oval in the middle where the carbon isotope values go up and the nitrogen isotope values go down. That is consistent with a period of starvation. But you can also see the nitrogen isotope values between the age of 9 and 14 increase. So maybe that's showing that then after that he actually did have access to more animal protein, and he obviously survived into adulthood. So this is telling us a little bit about the kind of his experiences when he was younger, and then how his diet might have changed during those periods of life. And then this is a woman who she died between about 30 and 40. She really sadly died in late pregnancy, so she actually had an unborn child. She similarly, we measured her oxygen isotope values, and again she had values that were too low for Britain. So again suggesting this either further north or higher altitude. And this is where we actually do have DNA from the Francis Crick Institute in London actually analysed, and her unborn baby, which basically means her as well, has ancestry from the present-day Italian peninsula. So this is suggesting that either, maybe the Alps or the kind of Apennines, which are the mountains in Italy, that would be consistent for where she was born. So, again, where did she, that's a long way to travel 1,200 years ago. We do know that people did travel that far, but it's kind of amazing to then find her, 6 miles down the road from Oxford, and thinking about her story and how does she get here. But the sulfur isotope value of her femur and her rib are both local to Oxfordshire. So she obviously spent a good portion of the sort of latter part of her life local to in Oxfordshire, and so she presumably moved at a relatively young age. Okay, so that's very much a whistle-stop tour, just to give you a bit of a taster, really, of the types of things that we can learn about people in the past. Just two of the aspects about what they were eating, where they came from, but just to kind of show you that we can really understand, we can start to reconstruct people's lives, and just build up a little bit more of this kind of in-depth picture of what life was like in the past. So thank you very much, and yeah, I'm happy to answer any questions if you've got time.